I couldn't hear all the introduction, so I quickly run over it again. So my name is Andreas Hollenstein. I'm scientist at Roche in the group of immunosafety, which is part of pharmaceutical science in PRED, and I'm located in Basel. So in this rather new PRED center, and it's actually a very nice place to work. So not only because of the science, but in the morning on my way to work, I cross the Wettsteibruck. So on the right side, I always see this nice morning impression of the so-called new skyline of Basel. And when I go back in the evening over the same bridge on the right-hand side, I see the stunning sunsets over the so-called old skyline of Basel. And actually, those two pictures are a quite nice analogy to my title. As you probably know, Basel consists of two parts. So there's Klein Basel and Groß Basel. And they're connected by this Wettsteibruck. And there's always like a bit of a competition, even they're part of the same city. Between those two parts of the city, kind of a rivalry between siblings and so on. And I think that's a nice example what we also see with maps and in silico prediction. And with this unmasking, with this data I want to show today, I try again a little bit this Wettsteibruck and bridge in silico prediction and maps detection for immunogenicity assessment. Quick overview of what to expect in the next 15-20 minutes or so. Quick executive summary so you can get prepared for what's coming next, which will be a short introduction to MAPS for lead discovery. I brought you two cases I would like to discuss initial findings we had with mechanistic considerations, and then also make the bridge again to in silico considerations, wrapping up with some final thoughts and most importantly, the acknowledgements. I guess everybody's aware that MAPS, so MHC-associated peptide, peptidomics, has become a central tool for immunogenicity risk assessment for large molecules. I guess there's basically no company that is in large molecules that not at one or the other stage leverages on this very informative assay. On the other hand, we had the in silico prediction, and since its introduction for class 2 presentation, it was widely, or still is, considered over-predictive because when it was compared to MAPS data, there was quite often peptides that were probably predicted but haven't been detected by MAPS, which led to this conclusion. However, we have recently found some particular modifications of large molecules that had an enhancing effect on peptide presentation that could be measured by MAPS. And here I would like to showcase what's the impact of this enhanced presentation on the concordance between MAPS and in silico prediction for the presentation of class 2. As mentioned, at one point of molecule development, MAPS will be leveraged as a important in vitro assay. So if you start at the very beginning, it makes sense to use in silico because of its low price and low demand on resources. So it can go very high throughput, basically funneling a big discovery panel and shaving off like the binders that potentially have the highest propensity to be presented by class 2. Then at lead optimization, we can probably zoom in further, already leverage with first in vitro assays, looking probably on peptide levels. So I would say this is really mainly for lead optimization rather than for a screen, even if some of my colleagues would probably not contradict, but I still think for lead optimization, this is the most suitable for peptide simulation assay. And later when molecules are available, which is kind of a prerequisite of the MAPS assay, which also probably puts it a little bit later in the development stage. So in the lead selection, we can use MAPS and other PBMC-based assays, for example, with T-cell stimulation assay to further refine the immunogenicity risk assessment. So MAPS is therefore a central tool for sequence assessment and the assessment of immunogenicity risk. However, as all assays, it needs careful interpretation of the found data. So there's quite often the notion we found T-cell epitopes and in the MAPS assay, which is probably a little bit going into much details, but presentation doesn't necessarily mean it's a T-cell epitope. We still need the cognate TCR detecting the peptide presented in class 2. And as for many assays, but I would say also particularly for this, I think I never said more that absence of evidence doesn't equal evidence of absence in this kind of assays. And why that so is, I will also touch on in the next slides. So jumping to the first case, for those who are not familiar with the methodology of the MAPS assay, so the starting material are PBMCs. Then monocytes are isolated with magnetic beads on CD14 selection. Then they will differentiated into immature monocyte-derived dendritic cells. They will be then incubated with the asset, with the large molecule. The idea being that these dendritic cells, mature dendritic cells, will take up the protein, process it, and present it on the surface. So once that has happened, the cells are getting lysed, and the class 2 molecules are pulled down with immunoprecipitation. Then the isolated class 2 molecules from the peptides that have been bound will be eluted, and those peptide mix will then be analyzed by LC mass spec. And this then basically tells which peptides have been bound to the class 2. And we like to visualize this in those heat maps. So here's a classical heat map of an IgG1 molecule. Each line here basically represents one donor. So we normally run it with 10 donors. And if we see here those clusters popping up, those were peptides. For example, here framework region 3 has always pretty strong peptides that they bind to class 2, highly preserved for most IgG1s. Those are actually always detected, and this goes all along the sequence covering constant and variable region of the molecule. In the same run, interestingly, we had the very same monoclonal antibody, but then fused covalently to a biological payload, and we are pretty sure there is no receptors on dendritic cells that would recognize this payload. However, the effect was quite impressive. So what we see suddenly from this very same antibody, suddenly the heat map is glowing up compared to the parental antibody. So really we see new clusters appearing, and also the magnitude you could appreciate a little bit the intensity of the colors gets higher. So we have more peptides detected, and also the magnitude of the detection of the antibodies was highly enhanced just by fusing this moiety to the monoclonal antibody. So now the head scratching started. What could be the underlying mechanism for that? And oh no, first I forgot that we actually were thinking, is this just a coincidence? Kind of a combination target with payload, but then we used another monoclonal antibody, which also has this standard map spectrum, and fused it to the very same payload, and we got the very same effect of new clusters popping up all over the molecule. So there's not a certain region that was preferred, but it's really overall again constant and variable region of the molecule where presentation was enhanced. And now the head scratching started, because what could be the underlying mechanism for that? And for this we deployed an assay that has been established by our colleagues in Pennsburg. So it's called the DC internalization assay, in short DCIA. It starts off pretty similar as MAPS. So we also take monocytes from PBMCs by positive selection on CD14. We keep them in an immature stage because then the uptake is enhanced. We take our test item and label it with a pH-sensitive dye. And the idea is once this, if you incubate this labeled antibody or molecule with those immature dendritic cells, they will be taken up and then they get a pH drop in the lysosome, and this can be detected in the readout by flow cytometry. We also engage a quality control just to make sure that the DARs of the labeling are comparable between molecules, and also to see if there's any unwanted specimens like aggregates or split products and so on present. But in our case, they all passed this QCs. And then here are then the subsequent results of this experiment. So again, we have a baseline uptake. It's an arbitrary scale that has been established using an antibody and a modified antibody, a derivative that has a high uptake. So that's where this arbitrary scale between 0 and 1 was generated. And you can clearly see that if we use the IgG1s, the parentals, they really behave as expected from a monoclonal antibody in terms of measured uptake in this assay. And as soon as we add the payload to this, we also see enhanced uptake. And again, we're pretty sure that there's no direct interaction of the dendritic cells with a receptor or any kind with that payload. And the question was also, do we, and this actually got even more clear when we give the payload alone. So if you would say, yeah, the payload probably has an effect on DCs, we also should see this effect. But if we uncouple the payload and give it separately, the uptake is not affected. So it's really required to have a covalent link between this payload and the antibody, which then, yeah, the picture would be it really pulls in the entire molecule into the dendritic cells. Another way to show that, I think it's always nice to answer the same question coming from different directions. If you look again into the MAPS data, so what we did here is we have an absolute peptide count, which is also kind of an internal control. So these are all peptides, also the endogenous peptides that are presented from our dendritic cells. And here we could see if there's any differences in the degree of presentation between if the agent or the test article would have an effect overall on presentation. But here we again see that the amount of peptides detected between the antibody and the antibody with the payload is basically unaffected. However, if we specifically look at peptides that originate from the monoclonal antibody, we again see that without payload, these counts are drastically lower than if we take the peptides that are together with the payload. So again, nicely correlating with the MAPS heat map data that we showed previously. So again, indicating we didn't like supercharge the DCs overall, but it's really specific increased uptake of this molecule due to the payload. So what are the consequences for correlation for in silico? For this, we had the big support from Morten Nielsen from the Technical University in Denmark, who's the father of probably you know the net MHC in silico prediction tool, and yeah, he was generating for us those diagrams. And what you see here is for the heavy chain and the light chain of our monoclonal antibodies, a spectrum that would basically represent here for the MS data to start with. So what we saw before on the heat map is now here in a linear fashion, probably the best to illustrate on the light chain. So if you follow the blue line, so here we didn't detect any clusters in the VH region at the N terminus and some clusters here at the C terminus. And if we take the MS spectrum of the monoclonal antibody with the payload, we see that here are those new clusters that were then newly discovered when we have this enhanced presentation. Similar happened here in the heavy chain. So this basically just represents the heat maps on a linear scale. It's also averaged over all the 10 donors. And what you probably can yeah, it's a little bit small, but you also see the magnitude. So we have different axes. So those for the enhanced presentation here go up to 100, 140, whereas here we're still at 35. So again, indicating the increase in magnitude of the presentation. And now if we look at in silico data, we just take again the same mass spec spectrum here that we had from the enhanced one. And for example, here in the region around position 100, we nicely see now this concordance between in silico and the mass spec of those new peaks, which in the blue line we wouldn't have. So again, making the point that even though, yeah, for the monoclonal, those clusters were not detected, if we enhance the presentation, we suddenly get the signals, and they are actually in agreement with what previously was predicted by in silico. However, on the other hand, we also see that new peaks are coming up that are not mapped with the in silico. So there we're still a little bit puzzled what happens there, but that's still under investigation. And more a visual type, but we also made the numbers, the correlations. So what we can see here, that basically the concordance or the correlations for the antibody with the payload in general increase compared to what we see for the monoclonal antibody. Here it's probably, again, there's like two counteracting forces. On one hand, the concordance increased because, yes, we see the clusters that now nicely correlate, but we also pull down basically the correlation if you have signals like this. So, but on the other hand, from a safety perspective, it's always better to be a little bit, better immunosafety than sorry. So from that perspective, that wouldn't be much of our concern. Okay, case 2. Similar starting point. So again, we have a monoclonal antibody with a typical presentation as we would expect from MAPS. Here, probably, as you can also see, the sensitivity seems to be higher. That's also probably, we have this variability with we get a new instrument, you get new software and so on, and suddenly you also see new signals. But again, if we add a payload here, and for this payload, we know there's a direct interaction with a receptor on the DCs. We again see this enhanced heat map. So again, more clusters coming up, and also the overall intensity has increased. And if you again deploy the same assessments as previously, the DC internalization assay, same picture. So the monoclonal antibody alone here performs like the low accumulation control, probably a bit higher, which could also explain the enhanced signals that we saw before. But clearly, if we have this payload added, the signal for the internalization drastically increases. And also, if we look with the MAPS data, the absolute peptide counts are not affected. So again, we don't have a direct pharmacological effect of the payload on DCs, because independent, if or without, they are presenting the same amount of endogenous. But again, if we look at the delta between antibody and versus antibody with the payload, again, we see this clear enhancement in signals, similar to the case we had before. If we now look at the in silico data, and here it gets a little bit wild, in this situation, we were precipitating all HLAs, so DR, DP, and DQ. So there was a pan precipitation, and that's what we kind of also now looked if that impacts the data. And to start with, again, similar diagrams for the spectrum of presentation on a linear scale. So here in green, we have what would be predicted for DR only from an in silico perspective. And here, what would be predicted for DR, DP, DQ, and for the alleles that were from the corresponding donors we had in this assay. What's interesting to see that those profiles already do not change much. So we see probably here for this purplish line that now here shows some clusters, but still the contribution of DP and DQ seems not that big. And if we now look at the effect of the enhanced presentation, we already have quite a good coverage to start with. So those peaks are pretty well captured already by the monoclonal. But for example, here at this position, we already see that the mass spec for the monoclonal antibody didn't detect any clusters, whereas here then with the enhanced presentation, we slowly see also here a peak shaping up. Again, also taking the magnitude into account, because we have much more peptides, gives again a higher confidence that these are real clusters. But also we see again these clusters coming up that are not mapped by the in silico. And here, if we go for the numbers, for the correlations, the effect is not as strong, because we actually already start with quite a good correlation to start with for the monoclonal antibody, which is then not further enhanced or even slightly decreases, because probably we get new clusters that were not mapped. But for Spearman correlation, for example, we have quite a consistent increase of the correlation, probably because it's a non-linear correlation probably in this case. So overall, again, we find clusters that haven't been previously found, and some of them matched in silico, but again, we also pull down the correlation by those new clusters that we cannot map on the in silico. So yeah, to wrap up, as mentioned, MAPS has become a central tool for immunogenicity risk assessment, and the predictions are considered over-predictive because they couldn't be mapped to MAPS clusters. And I mean, that's more of a joke, but sometimes it's the notion that in silico doesn't predict with correlate with clinical ADA rates, which I guess, I don't think doesn't need to be the case. I guess there's not a single assay that correlates with ADA rates. It would like asking correlating affinity with overall response rates, and nobody would have this idea. Our findings suggest that most, I didn't know what to put. I put most false positives from the in silico predictions remained just basically under the detection radar for the MAPS assay. But in the future, potentially MAPS findings and in silico predictions will converge if we get higher sensitivity of the machines. For example, more sensitive mass spectrometers, enhanced data analysis methods. So we had examples where we took the same data set with a new software analyzed, we got new peaks, and those new peaks also were already predicted by in silico. So there's also still quite some ground to cover there. And I guess now the question a little bit, which is in the room. So how different map sensitivities can affect the risk assessment or sequence optimization effort. So and reporting, for example, if there is already efforts ongoing that we should have more preclinical data reported for IND filings. So again, very bluntly, if you have a dull assay, you basically have less findings, and so it's makes sense not to get too ambitious with your map sensitivity, but we know that can't be the way. So there should be ways, if we have to use the data, that we use them in a way that makes that is sensitive and is really useful, and not just reporting it for the sake of reporting. And there could be possible alignment tools, standard compounds, but we all know the standardizing assays or distributing such compounds and so on comes with its own challenges. So that might be not the solution for everything, but at the end, as usual, we just should be critical with looking at those data. And with this nightly impression of our campus, I would like to acknowledge all the people that were involved in this presentation. So it's from our Roche Innovation Center in Basel, from PS, all the people that helped support me with performing the experiments, interpretation of these experiments, and also conducting the experiments in the 360 lab. The Roche site in Munich for the internalization assay work, our site in Zurich that basically contributed with some cases, and big shout out to Morten from the DTU supporting me with all the in silico analysis. And with that, thank you for your attention and happy to take any questions.
Thank you very much indeed, Andreas. So if there's any questions, you can walk up to me. I'll pass you the microphone. Yeah, we do have a question, Andreas, for you. You can walk up. 你可以走过来这里啊，因为他这里的麦克风，这边小心点声。我们也是打开相机，所以你可以跟对。
Hello, thank you. Very nice data. And I have a couple questions for you. First, I think the payload thing is particularly interesting. First is, can you tell us a little bit about the nature of the payload? What is that a peptide with a special sequence or whatever? And second, what is the working mechanism for this payload and how it works? And the last is, will this payload thing bring about any artificial effect for the map study and more map work? Because it seems like the payload will enhance the presentation capability in these cell assays. I'm wondering if any artificial effect will, or actual effect will be there. Thank you.
Yeah. Okay. I hope I will get them all. So yeah, starting on the nature of the payloads, probably I cannot tell too much as only that they are of biological nature. So they're not bigger than peptides for sure. And about the underlying mechanism, I mean, the second case, it's pretty clear. We're pretty sure that this direct interaction between DC receptor and the payload, because we know they match. For the first case, which is actually, I think, more intriguing, we don't think we have such an interaction, but all the studies on internalization that Thomas Kraft in his lab in Munich performed together with Michel from our lab point to the direction that charge patches are strong moderators of DC internalization uptake. And very simplified, the more positive charge patches, the higher the uptake. That's also how we designed those standard antibodies, by engineering in more positive charge patches. And then we see clear correlation with enhanced uptake. And we know in the first case that the payload has quite strong positive charge patches. So that would be kind of our mechanistic explanation why the uptake is enhanced. So that's more a biophysical propensity of the molecule, which then leads, as an end result, to increased uptake. And to your last question, the impact, I mean, again, we're here at the stage of risk assessment. So this clearly enhances the risk for the molecule, because, the probability of a potential T-cell epitope to be presented is higher. But now, the question is, again, it's almost philosophical of the molecules which do not show in the MAPS assay the presentation enhanced. We still cannot rule out that those peptides are presented in physiological conditions, given long-term treatments, repetitive treatments, and the immune system just needs more time, and it will at one point also probably find those peptides that were not detected by the map. So that doesn't mean that in biology they won't be there. And with this enhancement, we basically just have now a way to show, yes, they have the biophysical propensity to be presented. They fit into they are taken up, processed, and fit the class 2 grooves of those donors. And this enhanced presentation, if that leads, what's the net effect on immunogenicity risk, that's hard to say then at the end. So that, I think, I'd like to cite one of my colleagues, the truth is in the clinical pudding, and we don't have a clinical data on these assets yet.
Thank you.
Sure.
Hello, Andreas, it's Nick from ProImmune here. Do you have any sense whether this changes any of the sort of antigen sort of processing inside the cell? Does the enzyme sort of specificity for cleaving the peptides, is that affected by this payload beyond uptake?
I wouldn't know if we're already on the level of enzymatic activity. I'm also there kind of oversimplifying. I always think like protease is a protease is a protease, but well, or a proteasome in this case with 3 proteolytic activities. But to that point, rather that we see these additional peptides that are not mapped. I mean, we were still kind of debating because they're not predicted in silico. Does that really mean they are basically unnatural peptides that were not supposed to be presented? I mean, our current working theory is, or working hypothesis is that we overload the system to such an extent that I don't know if it's proteolytic activity or the loading itself, that it even starts to load misfit, because there's always some affinity. There's nothing black and white and zero. So that, but that we kind of trigger or enhance this misfitting that otherwise we would not see. So, but we couldn't say it's because of proteolytic activity or just because of, again, biophysical interactions, hydrophobic or whatever binding effects of the groove and the peptide on its own. But yeah, again, we think there is the system will make mistakes because of that. Yes.
Okay, thank you.
We do have another question for you, Andreas. So we invite you to come to the computer.
Thank you. Hi, Andreas. My name is Fan Liu from NeoX Biotech. Questions about varying the peptide as a payload, either in the format, say you put the peptide into C terminal of the light chain or N terminal of the heavy or the light chain, or varying the identity of the peptide, either removing its binding paratope to the receptor or completely a different peptide attached to the same IgG1. I was wondering if you vary these variables, either the format or the payload is binding to its receptor, how would that modify the enhancement or even the clusters?
Yeah, interesting thought. Unfortunately, we didn't have the time to do those experiments because our function is mainly assessing the risk. So we could not go then having a follow-up and really delve into, as you say, I mean, it would be interesting thoughts on in the first case, kind of play around with surface patches and so on, or see what kind of, binding sites, if that's kind of would impact I mean, binding sites, the fusion side, if that, for example, would impact the presentation. Unfortunately, we didn't do those experiments, but those were certainly very interesting follow-up experiments. Yes.
Yeah, thanks, Andreas. Nice talk. Thank you.
Pleasure.
We take one last question.
Hi, Andreas, Julia Rosenfeld, Teva Pharmaceuticals. I have a question. Have you done any additional immunogenicity assessment of these peptides that you found in maps?
Now I have to think. Yes, I think they went also, but just from the top of my head. So I think they also went, for example, through a kind of a peptide simulation assay, and now it gets really vague because I don't remember, but it's probably good for me also to make a follow-up. I don't remember that there was kind of a correlation between signals we saw there between, they would differentiate peptides that were presented versus those that were not presented. So it's I think the distribution of what you normally find in such assays in terms of frequency of findings was not biased by the nature of the peptide coming from, I mean, sure, if you have more peptides, you probably also find more positive peptides, but if you take 10 in one and 20 in one, and you still have a 50-50 distribution, that wouldn't change. I guess that's basically where your question a little bit comes from. So again, my memory is very, very vague on that because I would have to look into it, but certainly something I should do. But there was no obvious bias, let's say it this way. Otherwise, I would remember that's for sure. So therefore, I would say there was not couldn't discriminate in another assay between those peptides.
Thank you.
Thank you so much, Andreas. So yeah, so we will move on to the next part really. So thanks, Andreas, for your time. Yeah, very much. All right.
Thanks for having me.
Thank you.
And yeah, have a nice meeting.
Thank you so much.
Thank you. Bye bye.
And our